Hi friends, a warm welcome to my channel. My name is Pihu and I am a student. I am a new YouTuber and this is my first time you're watching my video or visiting my channel. Make sure you hit the subscribe button and turn on the notification bell to help me to get motivated and keep on bringing new interesting videos for you. So let's start our video. Today I'm going to explain the chapter Cell, Fundamental Unit of Life. Now, what is cell? A cell is a structural and functional unit of life. In English, scientist Robert Hooke discovered cell in 1665 while examining a thin section of cork under his simple microscope. Structurally, a cell is formed of these parts: plasma membrane, cytoplasm, and nucleus. Now, what is plasma membrane? Plasma membrane is also known as a cell membrane. Each cell is bounded by an extremely delicate, thin elastic lining membrane called plasma membrane. A plant cell, it is present on the inner side of the cell wall. The plasma membrane is made up of two layers of liquid molecules with protein molecule. It is selectively permeable membrane. Now, why plasma membrane is known as a selectively permeable membrane? Because it regulates the entry and exit of some materials inside and outside the cell. Functions of plasma membrane: it gives a definite shape of the cell, it protects the internal contents of the cell, it regulates the entry and exit of substances in and out of the cell. Cell wall. Plant cells have an additional protection wall outside the plasma membrane called cell wall. It is permeable and made up of cellulose. Functions of cell wall: it gives a definite shape to the plant cell, it protects the plasma membrane and other contents of the cell, it doesn't allow much of water to come inside. Cytoplasm. The protoplasm which surrounds the nucleus is called cytoplasm. It is formed of proteins, nucleic acids, carbohydrate, liquid, and certain inorganic substances. Many small organelles are found inside the cytoplasm. These are known as the cytoplasmic organelles. Cell organelles. So there are basically four cell organelles: endoplasmic reticulum, ribosome, mitochondria, and Golgi body. Let us study each of them one by one. Endoplasmic reticulum. The endoplasmic reticulum is a network of tube-like structures through the cytoplasm. Some parts are connected to the nuclear membrane while others are connected to the cell membrane. Endoplasmic reticulum is of two types: rough and smooth. If ribosomes are attached to it, then the reticulum is rough, otherwise it is smooth. Functions of endoplasmic reticulum: it increases the surface area of the cytoplasm for various metabolic activities of the cell. It helps in the formation of cell plate during the cell division. Rough endoplasmic reticulum is associated with the synthesis of protein. Ribosomes. They are extremely small, round structures. They are present in the free state in the cytoplasm or attached to the surface of the endoplasmic reticulum. Functions: it helps in the synthesis of protein. Mitochondria. They are small, rod-shaped organelles found in the large numbers. Each mitochondria is bounded by two membranes, outer membrane and inner membrane. The outer membrane is smooth and the inner membrane is pushed inwards, forming cristae called cristia. It consists many enzymes for the oxidation of carbohydrates. Functions of mitochondria: it provides energy for the vital activities of the living cell. Golgi body. It was discovered by Camillo Golgi. It is formed by a saucer-shaped membrane called cistern. Functions: they store, modify, and condense the protein synthesized in the ribosome. They form the cell plate during the cell division. Plastids. Plastids are double membrane organelles which are found only in plant cells. They contain chlorophyll molecule. These molecules are responsible for the protection of food by the process of photosynthesis. They are therefore called the kitchen of the cell. Like mitochondria, plastids also contain their own DNA. There are three types of plastids: chloroplast, chromoplast, and leucoplast. Leucoplast, these are the colorless plastids. They store food in the form of starch, protein, and lipids. Chloroplasts, they are the green plastids, and chromoplasts, these are the colorful plastids which are present in the flower and fruits. Now let us study about lysosomes, which are also known as the suicide bags. These sac-like small, spherical, single-membrane bound vessels which contain enzymes. These are formed by Golgi complex. Functions: they help in breaking down large molecules of cell. They work in different against bacteria and viruses. During starvation, lysosomes digest their own organelles. This results in cell death. Hence, lysosomes are also known as the suicide bags. Vacuoles. They may be small or large in animal cell. Vacuoles are smaller in size and lesser in numbers than in plant cell. Functions of vacuoles: they maintain water balance in animals. They work in osmoregulation. They store various substances including waste products. Nucleus. The nucleus is a dense, spherical, and oval organelle. It is bounded by a double membrane called nuclear envelope, which separate it from the cytoplasm. Numerous pores are present in this membrane to allow the transport of materials between the nucleus and the cytoplasm. Inside the nucleoplasm is a thread-like structure called chromatin. They are formed of DNA and protein. The chromosome contains stretches of DNA which carry information for protein synthesis. These stretches of DNA are called genes. Functions: the nucleus is the storehouse of genes. Genes transmit the genetic information from the one generation to another. Nucleoid. In some organisms, like bacteria, nuclear membrane is absent due to the absence of nuclear membrane. Undefined nucleus containing only nucleic acid is called nucleoid. Structure and type of chromosomes. A chromosome is a deeply staining rod like structure with a part that is usually constricted. Types of chromosomes. There are four types of chromosomes: metocentric, submelacentric, arcocentric, and telocentric. Metocentric. Two arms of chromosomes are equal. Submelacentric. If one of the arm of chromosomes is smaller than the other. Arcocentric. If one arm of the chromosome is considerably small than the other. Telocentric. When the centrosome occupied its one end of the chromosome. Here is the diagram of prokaryotic cells and animal cell. Now let us study cell division. New cells are formed from the older one is known as the cell division. There are two types of cell division: meiosis and meiosis. Mitosis. In this type of division, chromosome numbers remain same. One cell forms two daughter cells having same number of chromosome. Example for growth and development. Meiosis. In this type of division, chromosome numbers became half. In this division, four daughter cells are formed. For example, during reproduction. Now let us study the differences between prokaryotic and eukaryotic cell. The average diameter of a prokaryotic cell varies from 0.5 to 5 meter. It lacks true nucleus. Membrane bound or organelles are absent in the prokaryotic cell. Cell division by fusion. An example of prokaryotic cell are bacteria. Eukaryotic. Diameter varies from 1 centimeter to 40 mm. True nucleus in eukaryotic cells. Membrane bound organelles are present. Cell division by mitosis or meiosis. Example organisms. Let us study the blue box questions given in the NCERT. Who discovered cell and how? Robert Hooke, while observing a thin slice of cork under his cruel microscope, observed that the cork was composed of several box-like compartments. He named the compartments as cell. Why is the cell called the structural and functional unit of life? The cell thus has a basic structural organism. This helps the cell to perform functions like respiration, obtaining nucleic acids, and clearing of the waste materials. Thus, the cell is the structural and functional unit of living organisms. Next, how do substances like CO2 and water move in and out of the cell? Through diffusion or the selectively permeable membrane. Why is the plasma membrane called the selectively permeable membrane? Because it allows the entry and exit of materials inside and outside the cell and not all the materials. Next, can you name the two organelles we have studied that contain their own genetic material? They are mitochondria and plastids. If the organism of a cell is destroyed due to some physical or chemical influence, what will happen? The cell will automatically die. Next, why are lysosomes known as the suicidal bags? Because during the disturbance in cellular metabolism, lysosomes are capable of digesting the entire cell by releasing their own enzymes. This is why they are called suicidal bags. Why are proteins synthesized inside the cell? In the rough endoplasmic reticulum. Let us see the textbook questions of this chapter. Make a comparison and write down ways in which plant cells are different from animal cells. Plant cells varies from animal cells because first, the cell is larger in size in plant cell. Cell wall is present in plant cell. The plastids are present in plant cell and one large vacuole is there. In animal cell are smaller in size. The cell wall is present. Plastids are not present and more vacuoles smaller in size. How is a prokaryotic cell different from the eukaryotic cell? A prokaryotic cell is smaller in size as compared to a eukaryotic cell. The nuclear region is not surrounded by the nuclear membrane. Single chromosome is there in prokaryotic cell and membrane-bound cell organelles are present in the prokaryotic. Eukaryotic, they are larger in size. The nuclear region is surrounded by nuclear membrane. More than one chromosomes are there and membrane-bound cell organelles are absent. What would happen if the plasma membrane ruptures or breaks down? The protoplasmic contents will be dispersed in the surrounding medium. What would happen to the life of a cell if there was no Golgi apparatus? If there will be no Golgi apparatus, packaging and dispatching of materials synthesized by the cell will be stopped. Which organelle is known as the powerhouse of the cell and why? Mitochondria is called the powerhouse of the cell because it is the synthesized site of energy rich ATP molecules by cellular respiration. ATP here means adenosine triphosphate. Where do the lipids and proteins get synthesized? Inside, proteins are synthesized in the ribosomes attached on RER, while the lipids are synthesized in SER or smooth endoplasmic reticulum. What is osmosis? The process of movement of water through a semi-permeable membrane from the higher concentration to a lower concentration. Next question. It's the question number 8 from your textbook. Let's answer first. Water gathers in hollowed portion of potato B and C because there is either sugar or salt. Second, potato A is necessary because it is a control experiment. Third, because A is control experiment but potato cup D is boiled, the cell membrane is inactive, osmosis does not take place in dead cells. Thank you for watching my video. If you like the video, do like and subscribe to my channel for new videos.